Assalamualaikum and hi, I'm Nur Tasni binti Syamsul Asmadi, student ID 2025486618. Today, in this video, I'll cover two practicals from our TRCRC field trip. First, practical 1, calculating mean, variance and standard deviation across leaf measurements to understand natural variation within a species. Second, practical 2, using quadrant sampling to compare plant distribution and density between two forest sites and what tells us about the factors shaping biodiversity. Tropical Rainforest Conservation and Research Center or TRCRC was established in year 2012 to restore tropical rainforests and address the critical rate of biodiversity loss in Malaysia. Now let's look at practical 1. Practical 1 is about basic concepts of statistics and variation. Practical 1 have three objectives. First one is to calculate and describe the mean, variance and standard deviation. Next is to understand the concept of correlation. The last one is to determine the relationship between leaf length and width. For the method, we need to collect 10 leaves and measure the length and width of each leaf and calculate mean, variance and standard deviation. Then we need to record and tabulate the data on graph paper and repeat the same steps for two sets. This is the data for leaf length for set 1. After calculating, we got mean of 80.57 cm, variance of 15.91 cm and standard deviation of 1.330 cm. This is the leaf width for set 1. We got mean of 1.60 cm, variance of 0.1444 cm and standard deviation of 0.3800 cm. Next for set 2, the mean of leaf length is 18.16 cm, the variance is 4.2104 cm and standard deviation 2.052 cm. For leaf width of set 2, we got mean of 1.871 cm, variance of 2.0789 cm and standard deviation of 0.4806 cm. Moving on to set 3, the mean of leaf length is 15.14 cm, variance of 4.6804 cm and standard deviation of 2.1634 cm. Next for leaf width of set 3, the mean is 7.23 cm, variance is 16.9703 cm and standard deviation is 4.1195 cm. So this is the plotted graph of leaf length against leaf width of set 1, set 2 and set 3. The red dots represent set 1, the green dots represent set 2 and the blue dots represent set 3. As we can see on the graph, there is a positive correlation between leaf length and leaf width. As the length of the leaf increases, the width generally tends to increase as well, though this relationship varies significantly between the groups. For discussion, Practical 1 focused on understanding basic statistics such as mean, variance, and standard deviation using leaf measurements. We collected data from 3 different leaf sets, each from the same tree species. Set 1 had the longest leaf with a mean length of 18.57 cm. Set 2 and Set 3 had slightly shorter leaves, showing that different branch trees have different average leaf size. Set 1 also had the highest variance in length, meaning the leaf sizes in that set were more spread out. Lower variance in Set 2 and Set 3 shows their leaf sizes were more consistent. Overall, the leaf measurement data showed that leaf length and width varied noticeably both within and between the three sets, suggesting that factors such as species differences, leaf maturity, and microhabitat conditions likely influence the observed variation in leaf morphology. Moving on to post-lab question, first discuss the differences or similarity of the mean of length between different sets of leaves. Do the same for the mean of width. The answer is Set 1 has the longest leaf on average, while Set 3 has the shortest leaf. This difference may be different due to light intensity factor. For similarity, Set 3 has the widest leaf, while Set 2 and Set 3 have relatively similar average width. This indicates that leaves from Set 3 are generally larger compared to others. For question 2, the question asks the mean of variance, the meaning of the statement, and also the standard variation. The variance is the average of the square difference from the mean, and it measures the spread or dispersion of the data. A high variance means the data points are more spread out from the mean, while a low variance means they are closer to the mean. The statement means that there is natural variation in the size of leaf, even though they are from the same tree. This variation is quantified by the value of variance. Set 1 also shows the highest variance, indicating that greater variation in leaf length. Standard deviation is the square root of the variance. It also measures how much the values deviate from the mean, but in the same units as the data. This makes it easier to interpret than variance. The third question asks why is it necessary to have different sets of measurements for the measurement of leaf width and length. The answer is first, it may improve the accuracy and reliability of the results by providing a better overall picture of the natural variation in leaf size. Next, it is for better identification, consistent patterns in the data, and reduces the influence of any outliers or unusual values that may affect the results if only one set is used. Now let's look at Practical 2, which is about factors that affect the distribution and abundance of organisms. The objectives are to introduce simple concept of quadroid, to enhance the power of observation, to record the difference in plant distribution within the quadroid, and to infer from natural observations the possible factors that affect distribution and abundance of organisms. To conduct this practical, we need a quadroid size 5 meter times 5 meter. One quadroid used at each forest type. Plants within the quadroid were identified and counted, and we need to calculate the density. So this is the results of our quadroid sampling. What can be concluded here is this quadroid sample reveals high forest biodiversity due to its wide variety of unique plant species symbols. By counting the repeated symbols, researchers can calculate the population density and map the spatial distribution patterns of each specific species. For observations, we have identified 15 different species and also calculated the density. For discussion point, quadroid 1 showed high diversity with 15 different species in a 25 meter square area. The overall density was 0.96 individuals per meter squared. The dominant species is kasai with 4 plants, followed by Jelawi mentalun with 3 plants. The singleton pattern, which is 9 out of 15 species, had only 1 individual. So this pattern is typical of tropical forests, indicating high richness but low evenness. The presence of dipterocarps and heavy hardwoods suggests this is a mixed dipterocarp forest. We also found fast-growing and fruiting species like Orthocarpus rigidus and Ficus benjamina. These different species suggest varied conditions across the plot, such as canopy gaps, age effects, or changes in light and soil that allow different plants to thrive. For the post-lab question, first question define the term quadroid in practice. A quadroid is a square-shaped tool with a fixed area, commonly 5 x 5 meters, used in ecological surveys. It helps researchers observe and record the types and number of plant species or non-moving organisms in a certain area. Question 2 asks 2 basic requirements of all the quadroid techniques. The answer is quadroids should be placed randomly or in a clear pattern to make sure the data is fair and not biased. Next, the size and shape of each quadroid must be the same to the results are consistent and easy to compare. Next, question 3 asks what would be the advantages and disadvantages of using a larger or smaller quadroid to survey sedentary organisms. For larger quadroid, it is good to record more species, which gives a better picture of biodiversity, but it needs longer time to observe and can be difficult to manage. Meanwhile, for smaller quadroid, it is quicker to use and convenient, but it may miss some species. For question 4 and 5, the answer is shown in the video.